All right, I always like to start off by asking this question. When is the last time you walked through a chestnut forest? That might be a kind of a strange question, but you probably haven't walked through one. And that's really unfortunate because the American chestnut at one time was one of the most common trees in the eastern United States within its range, even Long Island. So if you see a scene like this in the mountains, Appalachian Mountains, you would have about 1 out of every 4 trees being a large American chestnut. But that is not the case anymore. We have lost the American chestnut. So what I'm going to do today is tell you about our research at the College of Environmental Science and Forestry, where we're trying to bring back this iconic species. It's 25 years of research with Dr. Charles Maynard and myself, but this has really been a group effort of over 100 researchers, from high school students to undergraduates to graduate students, postdocs, technicians, and colleagues. So it's been a long time coming. So why would we spend so much time trying to bring back the American chestnut? The American chestnut had many values. Sometimes people would say it was the almost perfect tree. It had an agricultural value. It produced a very nice nut crop every year. These nuts could be eaten by themselves, could be ground into a flour for baking. It could also be brewed into a beer, so you're able to actually make alcohol from these nuts. And the people in the Appalachians before the blight actually kind of thought of it as manna from heaven, because they were able to go out, these poor farmers, and collect nuts every fall and sell them to the market, and they'd be shipped to places like New York City and sold on the streets. So it has an agricultural value. It also has a social value. It's really part of our heritage. The American chestnut has kind of been fixed in song, as you just mentioned when I came up here. The chestnuts roasting in the open fire, that Christmas song. Also on Sleigh Ride, where you hear about the chestnuts going pop, pop, pop. But almost anywhere you go in at least the eastern United States and even in the western, you can always find a chestnut street, just like you'd find a maple street or an oak street. So it's really ingrained into our society. Probably the most economically valuable thing about the American chestnut, though, is its wood. The wood is very straight-grained, it's very rot-resistant, it's a fast-growing tree. I had my grandfather who used to sell antiques that were older than, that were built before 1830. He used to tell me, though, you'd never find a chestnut cabinet, or it's very rare to find one like this, because chestnut isn't very hard. Instead, on the outside, you'd have walnut or black cherry. But on the insides of this wood, in the drawers and stuff, that would be all chestnut, because it was so easy to work. Chestnut was often used in exteriors because it was so rot resistant, fence posts, shingles, and all things like that. I always tell people today, if the chestnut was as abundant as it was, everybody would be making their decks out of chestnut because you wouldn't have to pressure treat it. Now, as far as we are concerned at the College of Environmental Science and Forestry, we are interested in this tree because of its ecological value. Okay, the American chestnut was a keystone species. A lot of wildlife relied on this mass crop that it would produce every year. It was a very consistent mass crop. The oaks that have since moved into the places where the chestnuts used to be do not produce a consistent mass crop from year to year. Wildlife from bear to deer to several species of bird would fatten up for the winter. And you might wonder, why is there a fish on there? Fish aren't jumping up eating chestnuts, right? No, what happens is that the leaves of the American chestnut actually are preferred by a lot of the aquatic insects in the mountain streams over the oaks that have since replaced it. And since they are preferred and they grow bigger and faster, that means that they provide more food for the fishes, and therefore it benefits them too. Okay, so the American chestnut was sometimes considered the redwood of the East, even though they aren't as big as redwoods, but it is one of the largest trees in the eastern forests of the United States. This is the classic picture of some lumberjacks standing among a stand of American chestnut trees. These trees typically would be 8 foot in diameter, with some record ones reaching up to 12 feet in diameter. Would be over 80 feet tall, with some record ones going up to 120 feet tall. So these are very, very large trees, and in this particular picture you can see how they have this nice timber type. But the American chestnut was actually a very diverse species, and even though this is what most people think of American chestnuts, these timber types, it could also have a spreading form, such as this one that was photographed in the 1970s or 1980s in Michigan outside its natural range. So you can kind of picture that as, in the poem, Under the Spreading Chestnut Tree. You can get that kind of form. And the reason why I'm bringing this up is because when we want to bring back this species, we actually want everything. We want to bring back all the different genotypes of the American chestnut. So what happened? The American chestnut was, again, once one of the most abundant trees, but not anymore because of a introduced blight. As people start bringing in Asian chestnut trees from China and Japan, what they didn't realize at the time was they were bringing over all the microbes that were on the surface of those trees. And unfortunately, one of the ones they brought over was a fungus called Cryphonectria parasitica. This is the cause of the chestnut blight. And as you can see here, it was very happy to come to the United States. That actually grew that way. That's one of my technicians got that picture. So what this fungus would do is it would jump off of the Asian species, which were resistant because they co-evolved together, and get onto the American chestnut. The American chestnut, because it never had been exposed to this fungus before, had no natural resistance to it. And what the fungus would do is it would enter through a wound in the tree, oftentimes at a branch scar, and it would colonize that wound and sit there for a while, kind of living as a saprophyte off the dead material, and then it would change its physiology. Two important things happen. It starts producing mycelial fans, which act like a mechanical wedge to help pry itself into tissues. But ahead of that, it starts producing acids. And the major one is oxalic acid. Okay? And that acid actually kills the plant cells ahead of those mycelial fans. And it forms a structure what we call a canker. And this canker will continue to grow and eventually girdle that branch, choking it off, preventing all the flow of nutrients up and down that branch, so the bulb dies. Okay, this was a very, this is very devastating. When the thing was first introduced in this country, people start seeing hundreds and hundreds of chestnut trees dying, and they really started panicking. And you see headlines like this, and this is when it first entered this area of the country in New York. You see headlines like, "All chestnut trees here are doomed. Chestnut trees face destruction," and they were absolutely correct when they wrote this over 100 years ago. Okay, so you have the kind of a distinction here that the chestnut blight, we're very close to ground zero for the chestnut blight. The chestnut blight actually started somewhere on the, I guess that would be the eastern end of Long Island. It was actually first described in 1904 at the Bronx Zoological Park by Dr. Merkel. And within about 50 years' time, it has spread through the entire range of the American chestnut, killing somewhere between 3 and 5 billion of the large canopy trees that used to be in these forests. Now, the chestnut blight not only kills American chestnut trees, but also does a lot of damage on Allegheny chinkapins, a related tree, and actually kills Ozark chinkapins too. So those trees have been very much devastated by the chestnut blight. And one last thing I want to point out is that the blight also survives on oak trees. And the reason why I point that out is it doesn't kill the oak trees, but because it survives on oak trees, you cannot get rid of the blight. It's always going to be here. So if you ever want to restore the American chestnut tree, you have to get a resistant chestnut. Okay, something that can survive in the presence of blight is not going to go away. Okay, I love this picture here. It's kind of hard to see because this is an old black and white, but this is a picture of a chestnut forest at the Shenandoah National Park back in 1912. And what you see here is, if you look at all this light highlights here, that is the catkins, the male flowers of the American chestnut in bloom. Okay, so this is in June, and people used to say it was like snow in June, okay, with the chestnuts in bloom, because they were so abundant, and it turned the tops of the trees all white from the flowering. Okay, and you can see how big these trees are compared to the houses in the foreground. This is actually the same year, 1912, along the Long Island Railroad. And what you see here is the blight is spreading through. The tallest trees in the forest are being killed. okay? And you can imagine people back in those days that as they rode along this railroad, they would see mile after mile after mile of the largest trees in the forest dying, okay? Very, very devastating. So, with all that destruction, why isn't the American chestnut extinct? Because the American chestnut has a unique ability to stump sprout, and because the fungus that kills a tree cannot compete with the soil microorganisms. Those two things together allow the American chestnut to survive today. There's not billions of trees anymore, but there's still actually millions of trees, which is great for making sure we maintain some genetic diversity in a restoration program. So what happens is the American chestnut tree can get killed down to the ground by the blight fungus. It resprouts, it grows for a while, and it can actually grow quite a bit. This is actually from a coppice tree, but a blighted tree would do the same thing. This is one season's growth, 7 feet from a good rootstock, so it can grow quite rapidly. But unfortunately, the blight is stuck in this, what we call a Sisyphus-like cycle. And you all know Sisyphus, right? From the Greek mythology, the guy who was doomed to roll a rock up a hill, wanted to lose control, had to start over again and roll it back up. Well, the chestnut tree is doing the same thing. It's getting killed down to the ground, it resprouts, it grows for a while, eventually gets the blight again because the blight's not going away, gets infected, gets killed down to the ground, resprouts, and keeps going through that cycle over and over again. Eventually, it will run out of energy and that root will die. But it can go through that for quite a long time. Okay, and people have been working on ways to stop the blight or make resistance trees for a long, long time. So, after a century of work, what are the choices for restoration? And one of the things that you might see if you go to the web and you type in chestnut tree, the most common things you'll see are chestnut hybrids. Okay, that's where they cross a American chestnut tree with an Asian species because they are resistant. And they produce a tree that might be resistant to blight. Now the question is, are hybrids okay for restoration? And you might think, because this is a new slide I just put on, so I'm kind of working my way through it. American chestnut trees are quite different than the Asian trees in that they're about twice as tall, they're more cold hardy, the leaves are quite a bit different, the form of the tree is different. There's actually quite a few differences in there, but people don't think about that. They think, oh, it's all chestnut, so it's okay. So I want to give you another example of a hybrid. Anybody seen this before? People, I don't know who did this, but somebody in some zoos actually crossed a lion with a tiger. This is also a hybrid. They're both the same genus, Panthera. One's Leo, one's Tigris, and they've actually produced viable hybrid offspring that can actually breed. Now, these offspring are kind of cool, but would you want to take those in a restoration program out on the African plains? Or would you want to take those in the jungles of India for a restoration program? Probably not. So the question is, would you want to take hybrids of these two for a restoration program? Hybrids are perfectly fine as an ornamental yard tree or as an orchard tree for agriculture, but do you want to do that for restoration? Okay. And this is a little bit more about that. The most common hybrid out there right now is a Dunstan hybrid, a great tree. It is made from American chestnut crossed with Chinese chestnut, 3 different varieties, then back crossed the F1s, or intercrossed, I should say, with the F1s. And they claim this is the most widely planted chestnut tree. Again, a good tree for a yard or for an orchard. But I want you to look at something here. Look at the size of those nuts. That's more like a Chinese chestnut. American chestnuts are actually a lot smaller. And if you want a tree that's going to provide food for wildlife, you got to think about the wildlife that's going to eat that nut. Okay? You don't always want to have it as an agricultural product. You might want to have it so that the wildlife can eat it. And people are making hybrids from all different kinds of crosses here, crossing American, Chinese, Japanese, European, and Saguinii chestnuts. So they have all kinds of things out there that's available, but will these actually be good for a restoration program? Probably not. What you really want to do is make a tree that's basically identical to the original American chestnut tree, except for being blight resistant. And there's a couple programs that are doing that. One is called a back-cross breeding program that the American Chestnut Foundation is working on. We work very closely with them. And the idea here is they take their hybrid American chestnut, Chinese American chestnut. Now remember, these things have been crossed, and we know there's about 40,000 genes in these species. So you have 40,000 genes from the Chinese, 40,000 from the American, and you cross that, and then you back cross it to American to try to regain the American genotype and phenotypes. And they've gone through 3 generations, and what they end up with is a tree that's 1/16 Chinese, 15/16 American. Sounds pretty good. When they're doing this, they have to make sure that they bring along 3 resistance genes, because the resistance in Chinese chestnut is quantitative, meaning that no single gene can give you full resistance. You have to bring 3 along to get full resistance. And when you're doing that at the same time, you're trying to get rid of the unwanted traits. Now, this is actually a good program. People do this all the time in agriculture, but it's a lot of work, okay? And I was presenting this because I'm going to show you an alternative that we're doing at SUNY ESF. So I'm going to compare the breeding to transgenics, and that's what we're doing. We're actually doing genetic engineering. That's why this is something kind of unique. And to compare that, remember that chestnut has about 40,000 gene pairs, half from the mother, half from the father. And remember the 2 trees are quite different from one another. So, and also with the breeding, you have a tree that's still 1/16 Chinese on average. So what I like to do is to kind of let you visualize this is use a book example. Let's say you have a book, and let's say the genes in the tree represent the words in the book. Okay, so if you have 40,000 words, that's about a 190-page book. And if 1/16 of that is written in Chinese, that means about 11 pages or around 2,800 words are in Chinese. Is that important? Oftentimes it's not. I mean, you could have a Chinese word put in every which way, and you can still get the gist of the book, right? It doesn't matter. But if it's at a critical plot line, like whodunit, that might be important, okay? So, depending on where those words are or where those genes are is going to be important. Now, let's compare that to transgenics or genetic engineering. Still going to use the book example. I'm going to pull out this passage in the book. It was very exciting at that season to roam the then boundless chestnut woods of Lincoln. That was written by Henry Thoreau in Walden. And what we're doing is, instead of mixing a bunch of genes together and then getting rid of genes we don't want, trying to keep genes in we do want, we're just adding a couple genes at a time, okay? So it's going to be like this. In this whole book now, it was very exciting that season to roam the then boundless blight-resistant chestnut woods of Lincoln, okay? Nothing to get rid of. We just added blight resistance, okay? And that's what we're doing. So you have a tree that's basically 99.999% American chestnut. The difference is blight resistance. That's the kind of tree that will be perfect for a restoration program. Okay, so what genes do we test? Well, we could go after the genes from the Asian species, but the problem with the Asian species is that, again, it's quantitative resistance, and therefore no single gene will give you full resistance. So you have to actually stack them up. That's a harder way to go. But you can also get genes from other plants, because plants have defense genes, and we can use those for chestnut. So how do you select the genes if you're looking at other plants? Well, you have to go back and look at how the blight attacks a tree. Remember that it forms these mycelial fans, it produces oxalate. So is there some way that we can counteract that virulence factor, that weapon that the fungus is using against a tree? And so that's what we do. We look for a gene that will do the job, and we will always remember this. It's not the source of the gene that's important. It's the function of the gene. So we're looking for a function, not a source, okay? And we actually found that in this particular enzyme, the gene for this enzyme. The enzyme is oxalate oxidase, and it comes from wheat. And whenever I say it comes from wheat, the first thing I have to tell everybody, this is a non-gluten gene, okay? Wheat has about over 100,000 genes in it. 2 of them are for gluten products, and this is not it, okay? And what this does is it detoxifies the oxalate that the fungus produces. So this is the chemical reaction. You have oxalate here with oxygen, this enzyme. It breaks it down to hydrogen peroxide and carbon dioxide. Hydrogen peroxide the plant can use to make lignin and cell walls. Carbon dioxide the plant can use to make sugars, or it can just expel it as a waste product. So everything fits perfectly into the plant. Now the great thing about this is it's not a pesticide. Okay? It does not kill the fungus. It has no cytal activity. And why is that important? because if you don't put that selective pressure by killing the fungus, you do not put the pressure on for selection for overcoming this particular gene, okay? And what that does, it makes the resistance much more durable. And the interesting thing here is basically what you're doing is you're changing the lifestyle of that fungal pathogen, the Cryphonectria parasitica, from a pathogen to a saprophyte. You just heard about saprophytes in the last talk, basically living off dead material, okay? That's basically the same way it's living on the oaks, by the way. Okay, so when I said it doesn't matter which, where you get the gene, it's the function of the gene. One of the reasons why I say that is because this gene is in all kinds of plants, okay? Oxalate oxidase not only is found in wheat, we took it from wheat because that's the most studied one, but it's also found in corn, barley, rice, peanut, strawberry, banana, azalea, tomato, cocoa, sorghum, blah, 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 and on. Okay, and sometimes it's in the parts you eat, sometimes it's not. When I saw the report on the presence of it in banana, they were talking about the banana peels here. So I was wondering, well, does it end up in the part that you actually eat? So I took my banana from lunch, put it in our oxalate oxidase assay, and sure enough, this assay detects the presence of the enzyme by changing color, and it does change color here, just like it does in the wheat germ as air control. So it is actually present in the part that you eat. So the question is, when's the last time you ate oxalate oxidase? Did you have a banana? I saw them out there, right? Or did you eat anything with wheat? Did you eat any of those buns or anything? So, a lot of people are worried about, well, you're putting this in chestnut, and it's not normally in there, and I might eat it. Well, again, OXO is ubiquitous gene found in almost all cereal crops and many dicots. So if you were to eat this banana chestnut cake here, you could get the oxalate oxidase from the banana, you could get it from the wheat, and if you used our transgenic chestnuts, you can get it from that. So the question is, does it matter? Okay, it should not matter. Okay, so how do we get a gene in? You know, we got a gene, we got it identified. How do you actually get it into the plant? Well, we actually use a natural genetic engineer. This is a bacteria that has evolved a way to put genes into plants so that it puts a gene in to make the plant make a food just for that bacteria. Okay, and it's a really interesting bacteria. Just this past year, you know, we live in an age of genomics where everything's being sequenced. All the genomes of everything's being sequenced, it seems like. Well, they sequenced sweet potato this past year, and what they found out is all commercial sweet potatoes are GMOs, are genetically engineered. And they've been all genetically engineered by that bacteria, Agrobacterium tumefaciens, about 8,000 years ago. I don't think any of the corporations were around back then. But so it was not done by a company, it was not done by a university, it was just done out in nature. So every time you eat a sweet potato, you're eating a bacterial gene that was put in 8,000 years ago. So we're using that same bacteria. And instead of putting genes in it wants to put in, we basically put our gene in here with a selectable marker and put it into the plant. And when we do this, we have to do it one cell at a time. And so we have to regenerate a whole plant from a cell. And that's going to go into the next series of slides where how do you make a whole plant from a cell? Well, basically, it's like making a tree from a seed the hard way, because basically what we're doing is mimicking what goes on inside of a seed during germination, but we're doing it in tissue culture. The way you do that is, first of all, you have to get the right starting material. So we collect immature nuts. There's about a 2-week window in the middle of the summer where you can do that. You isolate those nuts. You aseptically remove the embryos, the zygotic embryos inside those nuts. They've been fertilized. And then you, and this is a very tough process. Only about 1 in 1,000 actually form what we call a somatic embryo culture. These somatic embryo cultures are basically cultures that the embryos bud off new embryos from single cells continuously. And so that allows us to put a gene into a single cell, and eventually that could be put into a whole embryo, which then could be grown into a whole plant. And we can maintain these cultures in the lab for about 9 years or more. Then we use our natural genetic engineer to transfer the genes. When we first did this, it took us about 24 months. We're down to less than 12 months now. This little reactor here has really helped us quite a bit for selection, so it cut down the time. So Basically what we're doing is going through all the different stages of development using different medias. Dr. Maynard used to say each of these is a graduate student project, each step there. And we eventually get shoots in a jar, and we can make a whole forest in a jar there. But then we have to get these plants looking like normal plants. We have to root them. Probably some of you have done rooting with plants before. And we end up with a plantlet. This is not ready to go out in the field yet. There's no root hairs on these roots. There's no cuticle on the leaves. So we have to go through a process called acclimatization, where we slowly increase the light, decrease the humidity, and get them in the soil, into the greenhouse, and eventually in the field. And we've had our trees out in the field since 2006, doing field studies with them. Okay, so that's how you get the gene in. Now we have blight-resistant trees. And here's something that's interesting about it. What we found out with these trees is, this is the most data I'm going to show you tonight, or today. This is a chart showing the amount of the oxalate oxidase being expressed, okay? In this line, Darling 4 has just got a little bit. This one has about 40 times more. This one has over 140 times more. And the reason why I'm showing that, I'm going to show you also this, what we call our leaf assay. This leaf assay predicts how resistant the tree is going to be. And this is American chestnut here. It's been normalized to 1. The orange is Chinese chestnut. And these green bars here are, again, these different lines. And what we found is, as you increase the amount of the enzyme being produced, you decrease the amount of necrosis, which means you increase the amount of resistance, okay? Which makes sense. The more enzyme you have, the more of this acid you can detoxify, right? So, and that's what we found. So, this next thing is really kind of cool. Let's see if I can get this to work. This is a time-lapse video over about 2 weeks of a small stem assay showing that we have resistance. This LS1 is a wild-type American chestnut tree, and it's a starting line for this Darling 215 and Darling 311. These have the oxalate oxidase gene. This is Chinese chestnut control. All have been infected with a fungus at that point, and we're just waiting now for something to happen. Okay, so what you see here, wild-type American chestnut tree, very susceptible to blight. Chinese chestnut survives. These are, again, clonal to this one over here, but they have the oxalate oxidase gene, so they become resistant. And this is just one little video of it, but we've done this over and over again. These are a group of wild-type American chestnut trees that we have inoculated. They've all wilted from the blight. They're starting to sprout at the bottom. These are our transgenic trees with the oxalate oxidase, surviving fine. These are the Chinese. About half of them, actually a little over half of them, also wilted. This is a very tough assay because we're using very small stems. You have to be very resistant. So we always say that our trees seem to be as resistant as the Chinese chestnut, maybe even a little bit more. Now, the interesting thing is that we're not killing the fungus. Remember that. We can actually take those surviving American chestnut trees and take their little wound sites, and we can actually isolate the fungus back out from it, as shown here. So just like we do with the Chinese chestnut, so we're not killing the fungus. Instead, the tree and the fungus now, they are coexisting. And if we have any teachers in here, here's something for your class, if you teach young children. I don't know if any of you know, Charlie Chestnut is the mascot of the American Chestnut Foundation. And this is a little cartoon we put together to explain what's going on. Basically it goes, Charlie pleads, Jenny, this is Jenny Jean, we need your help. Buster Blight is using this acid attack, and the American chestnuts are losing the battle. Don't worry, Charlie, I made an oxo shield to protect you. Now the battle continues, but Charlie uses his new defense. Buster exclaims, oh no, I can't penetrate your shield. Sigh, does this mean we'll have to learn to live in peace? Absolutely. You have to, okay? So now we have trees, and if we were just doing a breeding program, a hybrid breeding program, or a mutagenesis type program, we could give those trees out right now. But now, since we're doing genetic engineering, we have to go through the federal regulatory process, which is a good thing, because we're testing things like mycorrhizal associations, insect feeding, all different types of things like that. But so we get reviewed by the USDA, the FDA, and the EPA. It's about going to be a 3 to 5 year process to get through it, and it costs about $3 million. We've already spent about $6 million to get where we are right now. But that's what we have looked forward to ahead time. But once we get through this, here's something that's really neat that we can do. I'm going to try to do this real quick because I'm almost out of time. Since this is a dominant type of resistance, meaning that when you cross this with a wild type tree, half the offspring will inherit that resistance, we can actually rescue a lot of the genetic diversity that's still surviving out there. And that's important for a restoration program. And the way you can do that is take your blight-resistant tree, cross it to what we call mother trees. These are wild-type trees that would be killed by the blight. Their offspring could be collected, and from this particular cross, like this one right here, these offspring, half of them would be blight-resistant, and each one would have a different complement of the allelic composition of each of those parents. So you can actually recapture most of the genes, the alleles from that mother tree in a restoration program. Then you keep outcrossing and you keep building up this diversity, get local adaptation, and so on and so forth. And we can always tell which ones are the transgenic trees because we have a nice little 4-hour assay that you just throw leaves in, let them sit for 4 hours, and you get this ring if they have the enzyme, or they don't get the ring if you don't have the enzyme. Okay, so people always ask me, so what are you going to do, cut down all the forests and plant American chestnut trees? Absolutely not. We're not going to cut down any trees to plant American chestnut trees. There are plenty of places to plant American chestnut trees. For example, mine lands. They are forced to do reclamation projects. We can plant chestnut trees there. There's historic sites. There's private landowners that have old fields and stuff. There's plenty of places. But you must know that this is not a quick project. The American chestnut is not a weed. It does not spread rapidly. You have to actually plant them. And eventually, if you get enough of them, they'll start spreading on their own. And so this is a century-long project, and you really have to have organizations such as the American Chestnut Foundation to really get this going. We actually try to get kids involved in this. This was a youth conservation day where we actually planted some of our transgenic trees. This is on a permitted site. Once we go through the regulatory process, we won't have to put them on permitted sites anymore. But this one is one that's permitted by the USDA. And the great thing about this is that these kids here are leaving a legacy. So these kids that planted these trees can come back with their grandchildren. And when these trees are very large, they can say, I planted that tree. You know, quite a legacy. Okay, so do you think the American chestnut is important? Okay, so people always ask me, well, what can I do? What can I do? Well, here's the most important thing you can do is talk about the American chestnut tree. A lot of people have forgotten the American chestnut tree. You need to get it out, get it into public, talk to your classes, friends, family, social media, and talk about how we have a project that can help bring that back. Now, if you want to get involved even further, you can join the American Chestnut Foundation. And for anybody who wants to, I have some forms up here. And you could actually, if you have some land or something like that, you can participate in what we call the Mother Tree Program with Alan Nichols, who's the president of the New York chapter. And what we're encouraging people to do is plant the wild type trees so that we have some ones that will form the female flowers at the time we'll get our transgenic trees approved by the regulatory agencies. And therefore, we can start increasing this genetic diversity. And the last thing, I'm told I have to do this by my development officer. We're at a point where we're no longer getting a lot of money from research grants because we're not doing research anymore. We're actually doing production and regulatory stuff. And so we do have a site that you can donate if you would like to the project. Okay, so I'm going to end here with this quote from Dr. Robin Kimmerer, who's a faculty member at SUNY ESF. She recently spoke at the UN, and I like this quote from her. It says, "We humans are more than consumers. We have gifts of our own to give to the earth." And that's the way I view our chestnut project. You know, it's our problem. Humans brought this disease over here. So we should give back and try to restore this American chestnut tree. And so I'm going to stop there, and thank you, and answer any of your questions.
And we do have about three minutes for questions.
I'll keep this very short. I've received about 60 of the American chestnut seeds or nuts from Alan, and Girl Scout in Floral Park I've received another 12. The 60 are currently being grown out at the East Meadow, Nassau County Cornell Cooperative Extension, and we have programs to put about 50 to 60 of them around in Long Island. So if you want to do that, get in touch with Alan, and he'll help coordinate with us.
Yeah, and I'll emphasize, do get in touch with Alan, because I can't do all this myself. Because a lot of times if you email me, I'll just kind of forward it to Alan, if you wanted to be part of the Mother Tree program.
I'm wondering if you've seen any outliers or resistance developed in any of the wild stock American chestnut?
Okay, so the question is there any naturally resistant outliers out there? There are organizations. There's the American Chestnut Cooperators organization who are trying to find some of those outliers and then cross them with each other. I'm very skeptical if there's any resistant trees out there because you find a big tree. I found 18-inch diameter trees that are 60 feet tall, and they have the blight. They just have been kind of what we call escapes. They've escaped the blight for a long time. So I haven't really seen anything that really proves to me that there are some resistant trees out there. They say there are. There's been reports that there are, but none of those people will inoculate those trees with the blight to prove it. And I don't blame them. I'd rather keep the big tree than try to prove it. So there might be. It was a big population, but I haven't seen it yet.
Go ahead. Hi, I'm Enrico Nardone. I'm with the CTUC Environmental Association. Thanks, Dr. Nichols. CTUC is actually the Long Island representative of the New York chapter, the American Chestnut Foundation. And we inherited a project from Dr. John Patente, who 15 years ago started an effort to try to keep the Long Island genotype going. Dr. Powell mentioned that they stump sprout, but that does eventually end as the tree root weakens. And so what we've been doing is trying to find mature trees around the island and pollinate them and then harvest new nuts and plant them. And when we started this, we inherited the project from Dr. Patente, we had 10 or 15 trees around the island, and that number has shrunk. So starting last year, we've really tried to increase our effort to find new trees that are mature enough to flower. And then in late June and into early July, we run around the island collecting the flowers and getting up on ladders and pollinating the trees. So we'd love to incorporate your help into this and finding new trees that are big enough to be flowering, and we can try to find them and then add them to our process for the cross-pollination.
Yeah, I think this is extremely important to get locally adapted trees and bring the blight resistance into those trees so that we can try to keep the genotypes in the same areas. So,
Okay, one last question.
Just out of curiosity, the set of genes, the resistance genes that the Chinese chestnut possesses, how do they grant resistance to the trees?
Excellent question. How does Chinese chestnut provide resistance with their genes? That is an open question right now. We actually are collaborating with a group called the Forest Health Initiative to try to look at that. We have looked at over 30 genes so far, and about 6 of them look like they enhance blight resistance slightly. And we're still studying how that does it. Some of them, basically, one's like an acid phosphatase gene that helps take up phosphates from the soil. It might just make the tree a little bit healthier, and that kind of makes it a little bit better. Other ones have antimicrobial activity, so they might actually help kill the fungus in some way, or at least slow it down. But we don't know that yet. It's still an open question, and we're still trying to figure that out. It's not an easy thing to answer.
Okay, real quick.
Yes, it's not a question. I am also associated with CTUC Environmental Association. We have a table here, and we have a sign-up sheet for many of the CTUC programs, and you can sign up if you're interested in what Enrico was talking about. If you want to participate in that pollination of the chestnut trees, working on the Long Island genotype, you can sign up at our
Okay, great. And if you want to join the American Chestnut Foundation, I do have some things here if you want to grab them after the talk. So.
Okay, thank you, Dr. Powell.
Thank you.